And our first speaker is Jurjen Heij from the Spinoza Center for Neuroimaging in Amsterdam. And he's going to be talking about a needle in a haystack, deciphering laminar-specific neural activity with high spatiotemporal resolution line scanning at 7.0 Tesla.
Yes, thank you very much for the introduction. And I would like to thank the organizers and in particular Professor Niendorf for inviting me to talk about our work on line scanning fMRI. So this is the brain. So far, nothing controversial, but the brain is so unimaginably complex that it sometimes feels like this. All of us are just looking for a needle in a haystack. And one particular needle of interest is the organization across the cortex in different layers. Input from the thalamus is received in the middle layers and then relayed to neighboring areas through upper and deeper connections. And due to these different functions and the way the microvasculature is organized, the hemodynamic response function differs across depth. And the HRF is not the only property of brain function that differs across depth. At our lab, we're very interested in vision. And the main method we use to map vision is population receptive field mapping. And a population receptive field is encodes a particular portion of the visual field. So when I'm looking at the dolomites, there's different parts of my brain that encode different parts of this view. So this is an important property to understand. And also these characteristics of PRFs differ across depth. Specifically, the PRF size tends to be smaller in the middle layers of the cortex, which corresponds to the input, the relay of information that I just highlighted. And this is shown in fMRI, and it has also been corroborated by electrophysiological experiments. But to achieve and to probe these characteristics more in depth, we need a very high spatial temporal resolution. Sure, we can reduce a brain volume to a partial volume, or even a slice, as we heard before, to go down, to go up in spatial temporal resolution. But now we can go even further using line scanning. And with line scanning, we can go up to 250 microns in the laminar direction with a temporal resolution of 100 milliseconds. So why would we want to do line scanning? Well, we have a lot of voxels in the cortical ribbon. Instead of 1 or 2 voxels and having to rely on interpolation to get our 7 or 8 layers across the cortex, we directly sample these voxels and get their time series. Also, we have a lot of samples in our data, which allows for a more accurate estimation of the hemodynamic response function. Line scanning MRI was first done in rodents by Xin back in 2014, and generally consists of 2 saturation slices outside the relevant area of the cortex. And after that, the frequency encoding is set perpendicular to the cortex, and then the phase encoding is turned off. And this is really the key in line scanning fMRI. If you skip the phase encoding, you can really go down in your acquisition time. So with this sequence, they were able to achieve a spatial resolution of 50 micrometers and a temporal resolution of 50 milliseconds. And to see if the laminar origin of the bold response was due to these input projections, or whether it was a property of the microvasculature, they investigated 2 well-known circuits, mainly the somatosensory area of the forepaw and the motor cortex, and they found distinct onset peaks. So for the somatosensory cortex, they found a very early peak in layer 4, which corresponds to the thalamic input. And they found 2 onset peaks in the motor cortex in layer 2, 3, and layer 5. And this corresponds exactly to what to the canonical microcircuit that I highlighted before. It took a while for line scanning to be translated to humans, and one of the main reasons was technical difficulties and the requirement for a very high field. And eventually, my colleagues were able to implement a sequence that worked. And similar to Xin, we have our saturation slabs, and then this is our area of interest. We turn off the phase encoding, so that we eventually end up with a 2D matrix where we have the position of the line on the y-axis and time on the x-axis. And with this, they found cortical activation in response to a flickering checkerboard, where we also see the sort of standard finding that the bolt increases towards the surface. But the brain of humans is much more folded than the brains of a mouse. So if you do line scanning in a mouse, it's pretty easy to find a patch and stick that line perpendicular to the cortex. But for humans, that's a lot more difficult. And also with this type of acquisition, we don't really know what's inside of the line. We just stick it somewhere randomly in the brain, and we hope that the task we're applying actually elicits activation in this patch. We're not really sure about that beforehand. So we need to do something about the planning. And this is where we invested a lot of time in. And the approach consisted of 2 separate sessions with at least 24 hours in between. So we have high resolution anatomical scans and high resolution functional scans, based of which we reconstructed the surface and eventually find a point on the surface that we want to target with our lines. And this vertex has a coordinate in whatever software space you use. And vertices always have a normal vector, and these are per definition perpendicular to its vertex. So ideally, we want to place our line like along this normal vector. And that way we get the line perpendicular to the cortex and really harvest the power of line scanning across depth. Then the second session started with a low resolution anatomical scan to be able to register the first session too. And that way we could transform the information from the first session, the coordinate and the normal vector, to the line scanning session and acquire our lines in the area that we designated before. So to go a bit more in detail in that story, once we have my vertex, I'm already quite happy. But now I have a coordinate and a vector. So how do I go from a vector to something the MRI scanner understands? Well, we can make use of little linear algebra. We can calculate the angle with each scanner axis, so x, y, z, with the vector that we obtained from the vertex, and then create a set of angles that we can insert in our MAR console. But first, we have to, of course, apply the registration matrix that we had to our coordinate and our normal vector, and eventually we can insert it in the MRI console. So to confirm that we're actually targeting the patch we intended, we can make use of this registration cascade that I highlighted before. Because we have the registration from session 1 to session 2, all the way down to the eventual line scanning, we can also go back to the surface where the vertex came from. So let's say this blue dot is our target. We can project our line to the surface again, and then see that we're indeed targeting the vertex that we want. So that is nice that we at least are in the spot that we intend to. So then it's good to acknowledge that line scanning data is pretty noisy, and we tend to mitigate that by default using a custom implementation of Nordic. And one method that seems to be quite effective is anatomical component correction, where we take voxels of the CSF and the white matter, run a PCA on that, resulting in this power spectrum. So here you nicely see the respiratory frequencies and the cardiac frequencies. And if we regress that out of our line scanning data, we see that these frequencies are nicely removed, and we end up with a slightly cleaner time series. Note that these are a lot of samples, so it just looks really condensed. So how to do line scanning? Well, the crucial bit is to skip the phase encoding, which allows you to go down in acquisition time. And then we also need a very accurate planning. We need some sort of framework to really plan this line perpendicular to the cortex. And then we need to do some denoising with the custom implementations of Nordic and anatomical component correction. And then what can we do with it? Well, as I said, our lab is very interested in population receptive field mapping. And one way we do that is by sweeping bars across the visual field and try to model what the particular voxel is encoding for in visual space. So here we see when we flash this bar across this PRF, it lights up and then goes down again. And the way that works is that we start with a set of parameters, mainly the size of the PRF and its location on the X and Y axis. And we assess its fit with the data. Here we see it's a pretty bad fit. So we'll just update the parameters again to find a new fit. which is a bit better, but still not satisfactory. So let's just update them again until we find a very nice fit. And then we'll say, we'll assign this area of the visual field to the particular voxel that we're targeting. So we'll take this as output. And this allows us actually to, because this is done for all voxels across the brain in the first session, we can actually implement these characteristics into our criteria to find the target that we want. And if we do that, we know exactly what part of the visual field our target is encoding for. And if we have that, we can actually directly go in with our experiment and target that particular area in much more detail. So in the previous experiment, we flashed the bar across the entire visual space, but now we just flash it around that PRF. So it's a lot more focused around this PRF to really probe its characteristics. So this is what we found. If we look at the target PRF, we see a pretty good fit. That's great. And if we then look at the line scanning, we see a slight shift, but still a very nice corresponding PRF in visual space. And this time course is different because the design matrix that we put in is different. So instead of the PRF having to wait for every time a bar crosses, the bars are much more focused around this, so it leads to a different time series. And then because our line is perpendicular to the cortex, we can actually extract the PRF characteristics across the cortex. So in this case, we had, in this particular subject, we had 6 voxels across depth, and they all show very nice correspondence and overlap with the PRF that we're intended to target. And then we can also look at the PRF size across depth and then see that the middle, the PRF size of the middle layer is smaller than the PRF sizes of the deeper and upper layers, similar to what they found in the electrophysiological data and earlier fMRI data. So what can we do with it regarding population receptive fields? So we can re-identify the PRF that we intended to target. That is great. And we can also probe these characteristics across depth. But we can also go in more detail into this particular PRF. So if we can, for instance, gauge what is going on when the PRF is stimulated with stimuli that are bigger than the size it corresponds to, because that would lead to suppression and the response will go down. And we can see what the eventual goal would be to see what happens at a laminar level when that happens. So if we assume that the PRF that we have in the first session is an average across depth, and we know, oh yeah, that's very good. We know that the middle PRF is smaller in the middle layers compared to the upper and deeper layers. We can gauge we can simulate what the response would be of these PRFs in the middle and deeper layers. So here we see that the middle layer is being suppressed earlier than the superficial PRF. And then we can find a set of stimulus sizes that maximally disentangle these responses. Then again, insert these into our experiment. So we're really letting the target that we're trying to investigate guide the experiment, guide what our experiment looks like. And that is very unique to this type of research. And it's very akin to electrophysiological research as they do in the rodents or non-primate human, non-human primate experiments, where the area is known beforehand and then they stick in an electrode or a neuropixel or whatever. This has very, it has similarities with that type of experimentation. So this is then the output that you can get from that. So if you average across the events and the cortex, you can get an average HRF. You can also split the HRF based on events. And we see that a larger stimulus size evokes a larger bulb response. We can also separate the response across depth, where we see that the bulb magnitude decreases when going towards the white matter, which is a finding that's often found. And we can also find fits for each of the deconvolved HRF to see what our voxel tends to respond more to. But the eventual goal would be to split based on events and on depth, so that we see that we can appreciate the downward trend towards the CSF for all across depth and for all stimulus sizes. These results are highly preliminary, so we're still tweaking our experimentation to really try and disentangle what happens when suppression is kicking in. So what can we do regarding hemodynamic response functions? Well, we can really tune our experimentation based on the target location that we're interested in. And we see that this HRF varies across depth and response to varying stimulus sizes. So it's really acting as a sort of virtual electrode where you allows sort of bridging the canonical fMRI experiment with electrophysiological experiments, which could then lead to avenues for studying human physiology non-invasively. Code for this is available, documentations are available, and I would like to thank all my colleagues that help me out on a daily basis and you for your attention.
Yeah, thank you. Do we have any questions?
Oh, yes. Okay, thank you very much for that great talk. I wanted to ask you, so you are doing line scanning, I guess, for two reasons. One, as you said, it's kind of like electrophysiology. You just stick an electrode, you just have a line. The other reason is, of course, sensitivity, so that you can, in principle, to get rapidly an image encoding over two-dimensional spaces, it takes longer. And if you do line scanning, you're averaging along 2 dimensions, restricting your scanning time, and you can go high resolution along 1 dimension.
Yeah.
But suppose suppose you have enough sensitivity and you are able to do over V1 or wherever in the visual field you are interested in, say 200 micron isotropic resolution imaging. From the point of view of the scientific questions you can ask with line scanning, would that be more interesting, more informative?
So you're talking about sort of a slice with 200 micron.
You take a 3D volume.
Oh.
Yeah. Say covering all of V1.
So ignoring all laws of physics, we can just go in of whole brain and 200 isotropic? Or?
I didn't say whole brain.
Okay.
It's maybe whole brain, but let's say visual cortex.
Visual cortex. 200 micron. That would give you about 4 voxels across the cortex in V1 at least, because it's a bit thinner. So if you have something like that, then you can really do more laminar network type of studies than this.
And I guess, I'm also thinking interesting from the point of view of going, let's say, you just have one point across the lamina, but now you can go across,
the columns.
Columns.
Yeah.
Thank you.
I guess it would be possible. Yeah, you can direct this line to do columns, but then you would need to somehow find that area where.
But you did leave out the temporal resolution from the discussion. So I wouldn't even call your line scanning a virtual probe or electrode, because usually an electrode is just a pinpoint thing. You are getting much more spatial information than an electrode. So
in that sense, it's more like a neuropixel, because that really has multiple electrodes across the.
Yeah.
Well, I wouldn't downgrade the electrodes so much, because these days they have so many contact points along the electrode. They can do many contact points across.
Okay. So if there's no more questions, I think we.
Question from the audience.
There was one online. Okay.
So the question comes from Nikolai Pavlak. Do you think we can use a high-resolution HRF for studying the effect of disease on microvascularity?
Yeah, that's initially also what this project was set out to do. So something with sickle cell anemia, and then find what the HRF or what the hemodynamics are doing compared to the health controls. Yeah, it's hopefully somewhere in the pipeline in the future.
Okay, so no more questions. So let's thank the speaker again.